Hello, I'm TJ and welcome to my garden. Listen along as we explore the art and science of gardening. You can leave feedback for the show at 661-368-5177 or visit our website at tjsgarden.com. Hello and welcome to week 44. This week I'm doing something slightly different. I'm basically just going to focus on one segment, which is an interview. Trying to squeeze 3 segments into each weekly episode was proving a little difficult with my schedule and making things a little crazy for me, so I apologize. The show's going to be changed yet again, but this time only a little bit. Yeah, and this episode's kind of a fun one. I'm actually doing an interview with a friend of mine, Sebastian Cocioba, and he is a genetic engineer. He's self-taught. He'll tell you in the interview. His company is New York Botanics, LLC, and he's been doing a lot of really cool and interesting stuff. And I think this interview and the last one, to a degree, kind of show a trend in what I'm maybe doing in future podcasts. I don't know yet. It's still too early to say, but I do definitely want to highlight where I can open source and generally open citizen science type stuff with gardening, because I feel that letting too many of the technologies and devices and everything else that we utilize for our gardens, letting those be patented, copyrighted, put behind various barriers to use and to sharing makes gardening harder. I feel that happens in most fields. And so having as much of that be as open as we can is really awesome. Now, in his case, he's trying to open up genetic engineering so the common person can kind of access it a little more. So we'll definitely get that. It's kind of cool. In the last episode, we talked about the open source seed initiative. I'm still poking around to find more cool open source projects. I don't know how much of the podcast that's going to be, but it's a thread I'll come back to again and again, I feel. As well as just general horticultural practice and gardening. So anyway, here is my interview with Sebastian Cocioba. Hi, Sebastian.
Yes. Hi.
Hi.
How are you?
So can you explain a little bit about what New York Botanics LLC actually does and kind of how you started that?
Sure. Okay. So roll the clock back about 13 years. So Mother's Day was like on the horizon, and I wanted to get my mom a present. She loves orchids. Orchids are way too expensive. So I figured out that Home Depot throws out orchids like every Wednesday or so, the ones that aren't flowered. So I just like took them from the trash and then put them under grow lights with some hormones and forced them to flower. And at one point I even sold a couple back to Home Depot as like a mom and pop thing. But yeah, that was a fun little racket. And I realized that there is actually a demand for plant tissue culture in general. So I incorporated under an LLC. That gave me a little bit more legitimacy to just saying like, hey, I can clone plants for you, like in an alley somewhere. And the result of that was New York Botanics. So at first it was just a way to and then like word started spreading just by word of mouth, and I started getting a bunch of orders. So that just became my small business for a bit while I was in college. And and eventually I started doing genetic engineering at home, which led to me learning enough to do consultancy for plant engineering. So like universities or like private companies mostly would send me DNA, and I gave them back plants, no questions asked, in that sense. So it's kind of like a transformation services. So they would outsource that service to me so that they don't have to do it themselves.
Now, I mean, obviously the news has kind of gone on and on with what genetic engineering is or might be, and we all know that, science reporting in the news is always wonderful.
Mm-hmm.
Can you explain because my audience is mostly gardeners, so can you explain to them what a transformation is in sort of layman's terms?
Sure. Okay. So the term transformation is used when they're saying the ingress of a foreign bit of DNA or DNA from somewhere else into the plant. right? So you transform the genome into a different form, as in it just didn't have that sequence and now it does. Um, so it's weird. I mean, like science jargon tends to use a lot of words that mean something in common parlance, but then in that actual application, scientific application, it means something else. But it basically is put a gene inside the plant. The plant is now transformed. Yeah.
Okay. I just want to make sure folks knew that in case they didn't. And a big part of what you're doing these days is actually trying to make that more accessible to everybody else as well.
Yes, yeah, yeah. One of my key goals is to, because I learned all this essentially by myself. I didn't have much mentorship. I mean, the internet is a wonderful place, um, but because I didn't have much mentorship and I still got this far, I truly believe that anyone and everyone can do science and contribute meaningfully to the field of science. So my goal is to not simplify by dumbing it down, but actually have a curriculum of sorts where you can start from knowing nothing about biology and end up with a designed flower that you can call your own, and all the theory and practice along the way.
I mean, so I know there are two main technologies you've kind of been using for transformations. I know you work with the really coolly named Gene Gun and CRISPR.
Yes. Yeah.
Let's as I understand it, the Gene Gun is something that's a little less precise and actually, in a lot of cases, being moved away from, right?
Um, kind of. So there's two main transformation technologies.
Just because it's so cool sounding, can you explain to everybody what a Gene Gun is?
Sure, sure. Okay, so a gene gun or particle delivery system (PDS), it's formally called a gene gun, which I found funny. It's essentially a shotgun. No hyperbole, like literally a shotgun, using micro pellets of either gold or tungsten. I use tungsten because I'm broke. Um, and there's DNA wrapped around that tungsten ball, right? So it's literally like buckshot, but on the nanomolecular level. So these are about 1 micron in diameter, 1 micrometer, and you use an extract from salmon sperm called spermidine to bind the tungsten to bind the DNA to the tungsten, and then once it's armed, you use a high pressure chamber that bursts this little disc at a particular pressure, and it just like a pop gun, and it goes, and then just shoots it into the tissue. And that tissue can be anything. Because I work with plants in biosafety level 1 parameters, I normally just shoot leaves.
Yeah, I mean, a Gene Gun. I just had to have you explain that because a Gene Gun is such. It superficially sounds so ridiculous,
Yeah.
and yet it's actually a really practical tool. Like it works. It does a very practical, very real thing. But just when explaining the function of it, just makes it
Yeah.
sound so insane. It's like we're just gonna
Yeah.
fire a shotgun at a little bit of plant tissue and
Uh-huh.
hope the
And
DNA
the
sticks
center
where we want
gets,
it.
but like literally hope and pray, because the center of the shot is obliterated, right? Like you can literally see a halo of death. But around the perimeter is when the speed of the bullets, if you will, are just the right velocity such that it doesn't penetrate through all the cells, just like the top layer or a few, and it doesn't kill the cell, right? So we're really working with probability here, and you have to adjust the height of your petri dish with respect to the gun barrel. It looks like an espresso machine, and it's in this vacuum chamber. So you really don't have much to play with except for like centimeter increments closer or further from the gun barrel.
Mm-hmm.
And the gun barrel, the shot makes a cone, right? And that cone of obliteration, if you will, changes with respect to height. So there's a couple parameters, including the air pressure, the amount of shot, and then the distance from the barrel that you have to modulate. And the efficiency is 0.1%. So basically, 1 out of 1,000 impacts will actually stick. The rest will just kill the plant completely.
And of course, every sci-fi movie begins with the genetic experiment going horribly wrong. But what usually happens when one goes wrong?
Okay, when
Because
one
you
goes
say,
wrong, your plant just dies.
yeah, that's what I was gonna say. You said 1%, and the truth is, most of the other percent, most of the time, if you put the DNA in the wrong place, it doesn't do anything. It just kills. Well, it breaks something that worked before, maybe.
Yeah, yeah. So basically, the overarching goal of all of these technologies, and I'm going to explain one more, a little bit more, a natural one, for the same type of concept. But all of these technologies just focus on getting the DNA into the cell, and then once it's inside the cell, It's understood to some capacity, but not fully rendered into text. So we don't exactly know how it integrates. We have some idea. But the main goal is that plants have plant cell walls, and then they have their cell membrane, and you have to penetrate both of those, but keep the DNA intact and not kill the plant cell. So you can see how that probability drops to 0.1% or lower.
So basically, you're trying to get a sliver to go right through the nucleus and then hope that the plant's internal mechanisms actually try and, repair what it sees as genetic damage by sticking that in.
Yeah, and what you do is you want to cause a little bit of damage because the integration of your gene into its genome is through a process called homologous recombination, which basically is an Indiana Jones statue swap, right? Between a broken fragment on the genome and an intact template that just happens to be floating around.
I love the mental image of a tiny little particle carefully balancing two DNA strands on a pedestal.
Right, and then it just swaps it, right? So you basically have to the left, and the genetic sequence to the left and to the right of your gene of interest is exactly the same on the genome as it is on your fragment that's floating around because of the gene gun. And then the part in the middle that's different is the part that gets integrated. So as long as the sides are the same, it'll target to that region, and you basically just swap. You swap
okay. That's actually more precise than I thought it was. I didn't know there was like basically error correction on the sides to kind of get it to think it's the right part.
Yeah, exactly. So through the proofreading mechanism, we do that. Now that is mostly done in the chloroplast, not the nucleus itself, right? So the chloroplast because a chloroplast used to be a bacteria a long time ago, homologous recombination mostly occurs in bacteria. So chloroplasts have their own genomes. They have, just like how the nucleus has the genome of the plant, the chloroplast has its own genome.
Kind of like how our mitochondria have their own as well.
Exactly. So in the plant, you have technically 3 genomes: the plant core genome, then the chloroplast, then the mitochondria. And people focus on the chloroplast because then you get more chloroplasts, meaning more genes, more of your gene of interest can be expressed in the same cell. Because there's only one nucleus, but 100 chloroplasts or more. Yeah. So circles of DNA that self-replicate within an organism.
Gotcha. And then what are the other 2 technologies you've been playing with as well?
Sure. So the main method of transferring DNA into a plant for the last 40 years now has been agrobacterium. So it's if anyone, especially in your audience, has seen those giant tumors on trees, or crown gall disease, which I can almost hear certain gardeners just shudder a little bit. So the causative agent of crown gall disease is Agrobacterium tumefaciens. And basically scientists in the late 70s, I believe, they figured out that the bacteria isn't secreting plant hormones to make the galls. It's actually transferring DNA into the plant to overgrow and make tumors. So it's naturally genetically engineering as a pathogenic mechanism.
And that's the same mechanism they think may have contributed to how we ended up with sweet potatoes, right? That may have happened in the wild, and then we domesticated them from there.
Exactly. Yeah, because the telltale signs are one of them, the terminators. Basically, if genetics were a language, there's grammar and syntax, and a terminator is a genetic period. It ends every gene, right? It's basically a hairpin loop that allows the RNA polymerase, the thing that copies the DNA into RNA, to hit a brick wall and fall off. So imagine Pac-Man hitting this hairpin along this ticker tape, and then it just plops off. So that terminator normally is a telltale marker for Agrobacterium because it has a very particular terminator that works both in bacteria and in plants. So the sweet potato, if you do PCR, polymerase chain reaction, which is basically a DNA copy machine targeted to a specific gene. If you do PCR on sweet potato, you'll see the NOS, the nopalene synthase terminator, right there. And that's one indicator that Agrobacterium was there. It's like a signature.
Oh, gotcha. That's the only way it ends up there.
Yeah. So what scientists have done was remove the tumor-causing genes and replaced it with this landing pad sequence where special molecular enzymes that act like scissors can cut and then be glued, and then anything cut with those scissors can be glued together. So it basically leaves this little dotted line in a part of the sequence where you can add whatever you'd like to it, and then the Agrobacterium will transfer your genes into the plant.
That still works mostly with the chloroplast, right? Or is that?
No, so this one is nucleus only. That's the cool part. So
Oh,
this one really engineers the actual plant itself, but there's one huge catch. It's random. So where it puts your genetic statement, your sentence, is randomly distributed across the genome. And it could be more than one insertion. So you can't control where, but you can control, I mean, you can definitely guarantee that it will. Where the gene gun is 0.1% efficient, the agrobacterium is over 80%. So it'll definitely transfer it. You just don't have control over where. So you have to make a lot of plants and then screen for your gene activity or do sequencing and figure out where it is and breed it until it's where you need it to be.
and you were actually talking to me before about the myoglobin tomato project you guys
Yeah.
are working on, and that's actually been a problem with that one, right?
Yeah. So at the School of Visual Arts, where I was a scientific advisor, we had a biodesign competition, which is basically open to the art students and design students to do a kind of like speculative design about the future of whatever topic they're given. And during our stint at the biodesign challenge, we were given the topic of food. So they're like, where do you see the food in like 30 years? But not 30 like, oh, okay, it's 2040 and we have cooler phones, but like 30 Orwellian years into the future. Like, what's the future of food going to look like? And the students got together and we powwowed, and eventually they talked about veganism and the lack of protein in foods, or how we're going to combat the need for protein, or the alleged need for a large amount of protein. And so we basically came up with the idea of keep the steak, ditch the cow, right? So we wanted to get a tongue-in-cheek kind of joke of the beefsteak tomato, the famous beefsteak cultivar of tomato, but literally expressing beefsteak. So but reducing that to practice, we're like, we can't really engineer yet the connective tissue and everything in another organism. So we basically distilled what a steak is to a series of proteins, one of them being myoglobin, which is really tiny, and it's actually the most well-studied protein ever. It was the first to ever be crystallized and its shape known. Because what they do is they crystallize proteins using a series of chemicals, and then they shoot it with X-rays, and they see how the X-rays bounce off the crystal. And by tracing it back, you can figure out the shape of the protein. Because it's so small, it's smaller than the wavelength of visible light, so you can't ever see it. But X-rays are really, really, really tight. They're really narrow because their frequency is higher, so they actually are small enough to bounce off of structures that are smaller than visible light.
Okay.
So we chose myoglobin. Because it's basically when you grill a burger on the grill and it bleeds, that's not blood, that's actually myoglobin leaking out. Just like how hemoglobin in our blood, myoglobin, myo meaning muscle, globin meaning globular protein. Yeah, so we decided to pick that one because it's short and sweet, and you pay per letter. When you order DNA, like synthesized DNA, you order it from a company and you pay per base pair. So we got a deal for 15 cents per base pair. 300 base pairs, so do the math, and that's how much it cost. It was basically $100. And we converted the sequence of myoglobin from the language of cow to the language of tomato, because every 3 letters of DNA represents one amino acid, and there's multiple ways to encode the same amino acid. So certain organisms like AAT versus AAG with the same amino acid. So there's a thing called a codon bias, which really affects expression. So we had to find a table for cow and a table for tomato and swap them out. And that was basically truly synthetic biology, because that fragment, that DNA sequence, exists nowhere else on the planet. And then I taught a bunch of art students how to do the transformation process. And then they did all of that, and they actually knew what they were doing, which was the coolest part. Like, it wasn't just like, be my robot. It was more like, this is your idea, I'm going to teach you how to do it, and then you're going to do it, and then tell me every step what you've learned, what's going on. And so what's supposed to have been a speculative fiction ended up actually being made, and we got our DNA results like literally the day before the competition.
Did you ever get one that expressed it quite the way you wanted?
So because right after the competition, since we were already kind of like pushing the budget and the scope of the lab, because it's a bio-art lab, but it's a lab solely operated in an art school. So when you have that kind of facility, we're already under like the microscope, and you're like, why is there an art school with a biolab? So pushing the project more forward to actually analyze the proteins in-house was actually a really, really difficult part of that whole process. So I did some preliminary analysis, and we got something like 20% of the total soluble protein in the tomato was myoglobin. But it remained as a speculative project for a number of reasons. One of them, if you make anything and it's a food, it has to go through the FDA to be cleared. And that's millions of dollars and like 13 years of clinical trials because you just made a drug. It's not food. Yeah, as soon as you engineer food, it becomes more of a drug, and it has to go through the same human safety trials, even if it's just a tomato with a single edit.
Okay, well, that's cool. I mean, that's a project I knew you'd been working on before. I didn't know the extent of it, so I didn't know if that was something that was being pushed forward by anybody, but it sounds like it was just an art project. Still kind of a cool one, though.
Yeah. I mean, like, we were nowhere did the competition say we had to make one, but I'm just like, well, since we're here, and it's actually going to be fairly cheap to order it because we live in the wonderful future where $100 gets you a plant cow part. So we're just like, yeah, let's give it a shot. I mean, it was one of the fastest GMOs I've ever seen made because we had such a tight deadline. But everything, just the stars aligned, and we basically nurtured a couple of cells that were transformed. Because when agrobacterium attacks the plant, it only attacks the wounded areas, and so the rest of the tissue is wild type, like the ones that the natural cells. So we have to kill those off. So along with our myoglobin, we gave it a gene for herbicide tolerance, and then we put herbicide in the media. So basically anything that wasn't resistant would die, and anything that was resistant was also expressing myoglobin. It's like a filtering mechanism.
Was this the glycophosphate resistance, or?
So we used kanamycin, which is normally a human antibiotic, a pretty good one too. Because remember how chloroplast used to be bacteria a long time ago?
Mm-hmm.
They're susceptible to the same antibiotics as many bacteria. So if you kill off the chloroplast, the plant turns white, and the ones that are resistant will stay green. So it's basically green versus white selection. And you just take the green bits and nurture those until they turn into a whole tomato plant.
So you're not necessarily trying to, I mean, you are, but you're not really killing the other parts. You're just killing off their food factories so that once you detach them from the rest of the plant, they're dead.
Exactly, yeah. And as long as you maintain that selective pressure by the kanamycin being present in the media until the end, what you'll be left with is only cells that carry your gene of interest. Because if roots can form in kanamycin-laden media, then you got it. Because kanamycin inhibits protein synthesis, and proteins make up everything. So if it's unable to do so, it'll die.
So the last interview I did was with Craig LaHoulier. I don't know if you're familiar with him, but he wrote the book Epic Tomatoes.
Oh, cool.
Yeah, he's hugely involved in, like, he did the dwarf tomato breeding project where they bred out a bunch of strains of dwarf tomato from the couple that were available. He does a lot of work in heirlooms, actually, and he Seed Savers Exchange. But I bring it up because he was talking about how in a lot of cases, even when you're breeding, say, open pollinated varieties, you're wanting to have some trait that lets you identify in some way success versus failure. So like you'll sometimes use a plant with potato leaf foliage, even though you don't necessarily want potato leaf foliage in the end plant, but it's handy because they know the genetics of how that works. And they know if they cross that with this plant and they show up with this foliage or that foliage, they have to have the other stuff they want, or, a successful cross.
Yeah. So that type of like marker assisted breeding is what large like agriculture biotech companies do. Like folks like Monsanto, I guess now Bayer, when they were doing their trait stacking, because you want all of your specific traits to be on a particular chromosome or at least all together. So the chance of you losing those traits gets smaller and smaller. But in order for you to know where that trait is, especially when it's not a phenotypic trait, like you can't visibly see it, like with the potato leaf display, if you can't track it visibly or through other means, you don't really know, like you said, success versus failure. So a lot of the times the herbicide tolerance is just a convenient way because you can just spray it with herbicide and the ones that aren't, they die. But then because agrobacterium transforms the nucleus, the trait for herbicide tolerance will exist in the maternal parts and the paternal. So it'll be in the pollen. And the worry is that weeds in the surrounding area could pick up that and become super weeds.
I'm just trying to process what you just said. The chloroplasts, those actually do get carried on the pollen as well?
No, so anything you transform to the chloroplast are maternally inherited. So they don't show
I'm
up
sorry.
in the male parts.
You were talking about using the agrobacterium, which is nuclear.
Yeah.
I got you.
Yeah.
Okay,
Yeah.
yeah.
So if it's nuclear, it'll show up in every tissue, including pollen, and
See,
that
that's,
goes
yeah,
again.
because I was thinking chloroplasts were a lot like mitochondria in that they're only maternal. So in us,
Yeah, yeah,
mitochondria only
exactly.
go down the female line, and in plants, their mitochondria and chloroplasts only go down the female line.
Exactly.
Okay.
Yeah. Um, yeah. So that's why chloroplast engineering has become a really big focus, because one of the key deregulation steps is, will this spread elsewhere, right? So if it's not just constantly throwing pollen that's laced with your transgene, um, it's one step lower on the regulatory requirements. It still needs to go through deregulation because it's a foreign gene in the same organism. But the cost of it has gone down. I mean, the USDA and the FDA are working with biotech innovation companies in order to streamline this, because there's obviously a lot of public pushback, and for good reason, because it should be case by case, right? So just rubber stamping it, yes, GMOs are good or no, GMOs are bad, isn't fair for anyone. So that type of discussion drove the need for alternative transformation methods. And one of the most fascinating ones is called the CRE-LOX system, and it's basically a way to delete the selection marker after the fact. You can get rid of the extra baggage of being herbicide tolerant, and all you have left with is your gene of interest.
Well, that how does that one work? I know
So
we're
these are
probably getting way too complicated for home
no
gardeners, but
worries, no worries. I mean, I can keep it pretty simple, but basically, there's an enzyme called CRE recombinase that looks for particular sites called LOXP sites, and then cuts left and right of that, and then fuses it together. So basically, you cut out something that's bookended by the specific sequence, and then that enzyme also glues the two bits together, removing the whole thing. And the only thing you have left is the scar that we've realized that it's so small and so insignificant that it won't contribute to anything meaningfully. So you just have this tiny little incision, like the scar after a surgery, which is infinitely better than having an active herbicide tolerant gene in a plant that doesn't need that herbicide tolerance to begin with.
that's actually a problem. Is we've intentionally engineered so many plants for herbicide resistance, but we've found that that has consequences we weren't really thinking of, as far as animal, the insects building up a tolerance, or I'm sorry, in the case of herbicide resistance specifically, the other plants picking that up and then being herbicide resistant.
Yeah, because I mean the whole point of it is to gain resistance so that you can kill off the weeds. And ironically enough, herbicide tolerance actually lowers the amount of herbicide you need, because you can just spot spray directly where the root system is for the plants. Um, so that's a benefit, but at the same time, if it's agrobacterium mediated gene transfer, it'll be in the pollen. And there has been instances of herbicide tolerant traits being seen in the surrounding like flora, right? Through means that we still don't quite understand exactly how. I mean, like weeds have figured out a way to not really register the fingerprint of each unique pollen type, and just
Mm-hmm.
takes in spurious pollen as an advantage mechanism. And honestly, that's so clever, because that's how you really survive. Just take, you can't move, so you got to take in whatever DNA you can and hope that it integrates into something useful.
Oh, so a lot of the weed species are actually more willing to hybridize with close relatives than, say, other plants, basically. The ones that have sort of become major agricultural weeds.
Yeah, that's what's being seen. And like I mean, the evidence is kind of here and there. It's not 100% guaranteed that these things will occur. They're just like, how the hell did these weeds to the left and right of this cornfield become resistant? And the conclusion through deductive reasoning is that it must be the uptake, right? But not a lot of these studies actually sequence the plant, or they sequence it and they find the telltale sign of genetic engineering. But that same part that you're sequencing could have come from a virus.
The other side, too, is that a lot of the crops we grow that are, major agricultural crops are New World crops, corn, uh, tomatoes, potatoes. So they're
Mhm.
all New World stuff. So they all have relatives in North and South America.
Right, right. Um, and on top of that, they're also susceptible to the same viruses whose parts we've stolen in order to overexpress proteins as a tool, right? So, for example, the cauliflower mosaic virus, one of the key promoters that drive gene expression in that virus, is one of the most powerful tools for expressing any protein in a plant, like crazy. I mean, you can get grams per liter worth of any protein, to the point where half the protein in your plant is your gene of interest using this viral promoter. So that's a really powerful tool for genetic engineering, but also is the same exact part will be found on anything that's contaminated with CAMV, with cauliflower mosaic virus. So it's actually hard to deduce whether it came from genetic engineering or that part came from natural exposure to that virus. So, I mean, more and more parts are coming out, more useful things. Like people are moving away from viral parts and they're using native promoters that are just highly expressed in plants or totally synthetic ones. I mean, the field is advanced, but it's a little bit difficult to figure out what is a GMO and what is not if the trait's not obvious.
One of the big public complaints is that the worry that these things are going to be harder and harder, because some plants it's very easy to keep from crossing with a GMO, like a tomato.
Yeah,
Tomatoes 95% of the time cross with themselves, right? It
exactly.
self-pollinates just
Yeah,
before the flower even opens, so no problem there. But like corn, I mean, the exclusion zone for corn is measured in miles. So
yeah, exactly.
so you put some GMO corn down the street and now everybody's got GMO corn, whether they wanted it or
Yeah,
not.
exactly. And the biggest worry in terms of these is more of just like an IP theft topic, right? Because if the pollinators for corn up the farm, I mean up the road to a GMO farm comes to your farm, you now cross pollinate, and at that point you're in IP infringement territories, and that's where it gets really murky.
Yeah, well, I mean, there was a farmer a while back who I guess they found some of Monsanto's Roundup Ready corn in his field. It had naturally
Yeah,
crossed with some he had, but he had saved
yeah,
seed and regrown it.
yeah. So now, like the hot topic is like stealth engineering, for lack of a better word, where the only thing that changed is the trait you want, and everything else is gone. It's like right now we're leaving a scalpel and a tweezer in the patient's body and stitching them up, and that's not elegant, that's not efficient, and moreover, it'll carry over to unknown circumstances. So we want to get to the point where we can just put one gene that we want, or more importantly, just change the genes that already exist. That's gene editing, and that's where I'll get to with CRISPR and Cas9.
Okay, well, let's go ahead and talk about that a little bit too. So,
Sure. Yeah. Okay. Wherever there is life, there's a virus for it. And bacteria are susceptible to viruses too, but they figured out a way to defend themselves using a bloodhound that basically sniffs out the telltale sign of that virus and chops up the piece and then puts it in a library so it can remember, oh, that virus was this was a virus that attacked me before. I know how to cut it. Right? So it basically makes a library of short sequences, um, clustered palindromic repeats, which is basically the CRISPR acronym, of these small pieces of DNA that are fingerprints of old viruses. So what scientists have done was exploit that mechanism and turn it into like a bloodhound that you can give it a sock. Like if it's hunting a fugitive, this is the analogy. You give it a sock, and that sock is technically the sequence that you're looking for. So you can have one enzyme that can be trained to cut any sequence of DNA as long as it fits a particular parameter. And it's really good at just making a single cut, a snip. And that snip will heal itself, but it'll heal broken. So that's basically knocking out the gene. So right now we have a pair of scissors that can be programmed to hunt any sequence in any genome, with the caveat that it also sometimes cuts elsewhere.
okay.
Sometimes. Yeah. And that sometimes is a little bit irregular. And in order for you to because it's such a subtle change, it's just a couple base pairs, sometimes even just one base pair. You'd have to sequence the whole genome to very high coverage, basically over and over again, in order to find where else it cut, because sometimes it's not obvious. So we're not quite at the point of like hyper precision engineering where we change exactly what we want, agnostic of what the system is, I mean what the sequence is, and we only get that change. And that caused a big ruckus when the scientists in China decided to engineer 2 kids to be resistant to HIV using this mechanism.
Yeah, that's been in the news, and I guess there's debate over whether it was really done, but it seems like it might have been.
Yeah, but the issue is that when they sequenced it, they only sequenced 80% because that's the amount of coverage you can get from a single human cell. Because they were editing embryos, and embryos are only like a couple cells before they get implanted into a female. So you can only take so many cells to destroy so you can sequence it. So they only sequence 80%. And 80% of 3 billion is like 600,000. 600,000 left of DNA where we don't know whether it cut elsewhere. And so these kids are going to be born with that edit, and we don't really know how it's going to go because we can't predict where else it cut. So it's not quite there yet, but we really wanted it to be, so people decided to do radical science. And that's where it got ethically murky, and we're in this, like, the can of worms is opened, and now people want to do this.
Okay, and kind of talking to that, though, you're talking, a big part of what you're doing is trying to get more people familiar with this technology and able to do it. But doesn't that open the door to more of that kind of thing?
One of the main aspects is that it's not so that they can do it just to do it. It's so they can do it so they can be familiar with this technology and be able to make a rational argument as to whether or not they are for or against this. Because access to these tools, as hard as I would try, is still regulated to some capacity. So a lot of the techniques I'm developing is for community labs to utilize. And community labs are basically science labs that you can join with like a gym membership. And then under that, you have safety oversight, you have containment, you have all of the hallmarks of a standard university lab, but you have access to it as a private citizen. So in that way, you can bridge the gap between access, but also responsible engineering.
So the goal for you then is to get more of this community lab, more basically better equipped community labs to do this kind of thing, rather than, say, just have any random person be able to do this out of their backyard.
Yeah, I mean, the technology is already out, and it's already really accessible to the point where anybody given just a little bit of time. I mean, I'm self-taught. It took me like 15 years to get to where I am, but that's because I grew with the technology, and I also had no idea what I was doing. And now it got to the point where people can have access to very good training fairly cheaply, too. I mean, CRISPR systems and engineering in general is available at most community colleges. So it's not whether or not I tried to develop these tools won't necessarily make a person more or less likely to do these edits. This is more for educational purposes, so you know who to vote for. So you know how these things are going to affect your life. Especially like our generation's kids are going to be biotech natives. We're biotech immigrants. We grew in with the technology. They're going to be firmly implanted. When they have kids, they can choose what color eyes they have, what kind of diseases they're resistant to, as like a commonplace choice. And that requires critical thinking and familiarity with the technology. So getting into the hands of schools so that they can train students to understand what this technology can do is my like moral imperative at this point.
Kind of to roll back a little bit. I mean, we've gotten to some really cool science stuff, which is awesome. But it is a show for Gardner. So I did want to talk a little bit about you did a lot of cell culture before you did the genetic engineering. Yes. A lot of tissue culture, stuff like that. And you've told me before, I just wanted you to kind of reiterate for these folks, how was it that you were able to propagate some of the really hard to propagate orchids, ones that actually have to have a very specific symbiotic fungus to start their lives on?
Okay, so at first, what I did was try to learn a little bit about that symbiosis. Like, what is the fungus giving to the orchid in order for that to work? Is it the mechanical presence of the fungus being in the seed? Is it a chemical? And of course, it's a chemical, right? It's a particular plant hormone that helps with germination. And ironically enough, it's found in coconut milk, in like coconut water. Basically, young coconut water is rich in nutrients and phytohormones that the embryo that's developing in that nut needs to survive. And if you make a smoothie of charcoal, bananas, and coconut milk, you can germinate most orchids in vitro.
Yeah, I had to bring that back real quick before we went too far with anything else, just because you told me that before, and it's just such a weird thing to think of, that you just make a smoothie of basically those things and you can get those orchid seeds to germinate. Because in nature, it's like they're very specific to the fungus they attach to, right? Like they won't just if they land on a tree with the wrong fungus on it, they're done. I mean, these are mostly epiphytes, right?
Exactly, yeah. And my favorite orchid, the ghost orchid, that only grows in Florida and nowhere else in the world, has like a spawn rate of like 50,000 to 1, right? Because that fungus also doesn't grow on all of the same species of tree, and it's like dependent on weather, and it's in the Fakahatchee swamp in Florida. Next to a bunch of alligators, you see this beautiful orchid with no leaves. It's just a flower and roots. It's like a primeval orchid. And in Thailand, they have companies that just mass produce this stuff. You can buy a flask for like $100. They mark it up obviously because it's a rare orchid, but for $100 you can get something like 400 little seedlings in a milk bottle in a gelatin mixture of a smoothie with charcoal, banana, and coconut.
And you
Yeah.
say it doesn't have leaves. Is it because it's parasitic, or is it, does it have photosynthetic
Um,
roots like some fungi do? Or sorry, not fungi,
so
some orchids do.
yeah, so it basically relies on just enough photosynthesis from the roots themselves. So the roots just look like Phalaenopsis orchid, like nice thick green roots. And because they grow in swamp environments under heavy canopy, they're pretty shaded organisms. They don't need a lot of light. But the thing is, it takes forever for them to flower. And then once it does flower, only recently did they actually catch a video of who pollinates this thing, because it's got this really, really long pollen tube, and they were asked this nectar tube, and they're trying to figure out who has a tongue that long, and it ends up being the sphinx moth, which is a moth that has like a 2 foot tongue, something like that.
Mhm.
Like I might be wrong on the numbers, but at least a foot long. And then there's this like evolutionary war between the length of the nectar tube and the length of the moth's tongue, because you want it to be just a little bit too short, such that the moth has to work for it. And as it works, its antenna pick up the pollen.
And that's the one that Darwin actually had the theory about, right? That we just recently found out was true, where he's like, there has to be a moth with a tongue long enough.
Exactly.
Oh
Yeah.
yeah, I remember that. I remember hearing about that. That's a story that keeps getting brought up again and again, where he made this prediction based on seeing other similar orchids and how they're pollinated and how that one was structured. And he's like, this specific type of
This
moth
must
with
be.
these traits has to exist. And then we found the thing.
Exactly. And for the first time ever in like a 10-second video with night vision, because it's a nocturnal pollinator, you see this just like big moth roll up, sit there for 5 seconds, and then zoom off. And it's the first time ever we've seen the ghost orchid being pollinated.
Kind of like watching a hummingbird.
Exactly. Yeah. Yeah. It like does its thing in 3 seconds. It's anticlimactic, but then it does that. But yeah, so the coconut milk and the banana has been refined to now knowing exactly what chemicals are needed, and those chemicals have become a mainstay of orchid culture, such that like places like Phytotech Labs, one of my favorite places to buy stuff from. I don't get paid by them. I just really like them. They have like orchid micropropagation kits that have like banana powder and coconut powder. But they also have the actual chemicals themselves if you want to dive deeper into the modulation. So to answer your original question, is I started doing like grids of different concentrations of the hormones required, and eventually, after hitting my head against the wall a bunch of times, I found the right mixture for the different plants. And then I talked to Aaron Hicks, who has like the largest seed repository of orchids, and I just started buying a whole bunch and started bringing new crop to market. But then I got too many. I got like too many orders, and like the Grand Hyatt wanted this like every floor to have this orchid painting, which is basically like an orchid mounted on cork on a velvet canvas.
Oh,
So it's like
I've
a
seen
living
those.
painting.
They do that a lot
Yeah,
with Tlansias as well.
yeah, but they wanted like thousands, and I'm just like I'm in the third bedroom of my mom's apartment. I don't have enough space. So as soon as I got an order like that, I wish I had the capital to get a greenhouse to do it because it's a sweet deal. But there was no way for me to get a loan because I was 18 and I just made my company. So the bank's like, yeah, we're not going to give you a loan.
And
Please.
as hard as it must be to propagate, that actually wouldn't be a bad orchid to grow inside either, because it has really low light tolerances.
Yeah,
Yeah.
yeah. So like just getting the pipeline down, and once you have the right protocol and it works in your lab, it'll never change. It'll always be the same thing, and you can predictively make thousands of them.
And orchids actually, I mean, that just kind of for the fun of it, orchids produce tons of seed because of the
Yeah.
sort of long, because they basically produce a seed with almost no endosperm, at least these varieties, and they're entirely relying on these fungal relationships to nurse
Hmm.
the young through that first early stage, right?
Yeah, and the seeds last like only a couple days unless they're refrigerated. So it's, I mean, like if you've ever seen an orchid seed pod explode, it's like a cattail when you crack it, and it's just dust in the wind, just blows everywhere, which is a nightmare if you're working under laminar flow and doing tissue culture. Just
Yeah. Well, I mean, especially since a lot of the rare ones have the smaller seeds, where it's almost like a flowering plant desperately trying to create spores. Like
exactly,
they're
exactly, yeah,
minuscule. Uh,
yeah. And then I buy like a microgram of spores, which is like 10 of them. And I'm just like, where is my $20? Where did it go?
$20 for something you can't see.
Yeah. And you're just like, I hope that this dust is seed and not like dander from somebody's scalp, you know?
And I think this goes to show, because you live in an apartment in New York, right?
Yeah. Mm-hmm.
Your own apartment now. I think you said before you moved out of your mom's, right?
No, so I moved to college to do that, and then I moved back in because now I'm basically roommates with my parents, where I pay for the lab in my room, and then because they're getting older, I take care of them and make sure the house is good and stuff.
Gotcha.
So we're crashing together.
That makes the point even better.
Yeah.
So to anybody listening to the podcast who thinks they don't have the space or ability or whatever to garden, you're listening to somebody
Yeah.
who you said you incorporated at 18, right? Or 19?
Yes.
Okay.
Yeah.
So he incorporated 18. He knew nothing of gardening, knew very little of genetic engineering, and now he does both in a New York apartment.
Yeah.
So you
Yeah.
really have no reason to like listen to podcasts and watch YouTube videos and then not garden. Like,
exactly.
if they have a yard, they're 10 up on you. Like,
well, infinitely. I mean, I would gladly pay for a little square meter in somebody's yard to try stuff. But now that I shifted to petunia, my whole house is just going to be covered in petunias.
Yeah, that's your new project, right? You're working on.
Yeah, I call it flowers for everyone.
And that's one you were talking about, where you actually want to create a project where people can grow some, like transform a plant and then keep it as their own plant, right?
Yeah.
Like
so what I'm doing is I'm trying to work with regulators to kind of cut a deal to see in what conditions can a person take this home from their lab, from the community lab, in a way that's legal. Because if you plant it in your dirt and it's a GMO, that's the willful release of a genetically modified organism, which carries potential penalties of up to felonies depending on how it's perceived, right? So there's the legal ramifications as to whether or not an engineered organism can be planted in your garden. Right, so I'm just like, can we flower press the flowers and then dip it in acrylic? There has to be some way for the student to take back what they've made. It can't just exist in a greenhouse of some dusty lab somewhere.
And I'm curious, so is there any way to like ensure some kind of sterility or something?
Yeah, so recently there was a paper on just how far you have to go. Just how far petunias go to make sure that they can't self-pollinate. Right, so there's very few at all self-pollinating petunias. In fact, most of them are self-deadheading, so they don't even set seed. Like the one that I'm working with is from Proven Winners. It's the White Cascade, I think. It's a Supertunia brand because it's sterile, so it can't produce pollen. So even though I use agrobacterium as my test bed, it doesn't produce viable pollen. So it has one degree of more security from a biological perspective.
Is that one a hybrid?
Yeah, that one's a hybrid, and I talked to the folks at Proven Winners, and they don't want to tell me what it is, but I'm like, well, I sequenced it, so I'm just like, let's work with me here, because I'm literally the only person on the planet that has this sequence, and I'd like to distribute it for free, openly everywhere. But if they want priority for like a year in exchange for giving me free reign, such that the students who work with it don't get like their wrists slapped for taking an asexual cutting. Right, because this is under plant patent protection.
Yeah,
because it's a patented line. So, and it's protected against propagation via cutting or tissue culture. And if the students are using it as a model, they could get in trouble in that sense, right? Because
gotcha.
I could easily just go through a open pollinated heirloom petunia. In fact, that's what I'm doing on the side right now. Is I have 17 lines of white petunia, and I'm just trying to find which one is the most easily to engineer. What's the easiest to tissue culture, fastest growing? Yeah, because I know that they're going to be weird about it. I'm going to be like, okay, the genome's going to go public because I'm allowed to sequence it. Nowhere in your plant patent says thou shalt not sequence. And then it becomes a public good.
And just out of curiosity, is their hybrid a triploid? Is that how they got it?
Yeah, that's. So I'm trying to actually do some research on it. Because I couldn't actually find any good leads on the Supertunia brand. On the Supertunia line. So I was digging through some of the patents to figure it out, and they just vaguely describe it. I'm just like, this looks like every other petunia. How are these plant variety patents even enforceable? Right. But I'm going to be doing some more digging, especially now that I talked on a podcast to a bunch of gardeners, they're going to be like, you didn't do your homework. Bad. But honestly, I will try harder to try to figure that one out.
Well, I hope they listen to the podcast and complain about things you did versus seeing that I'm talking to a genetic engineer on the podcast and stop listening to my podcast. So I'm hoping that happens. Because one of my goals with the podcast is to kind of introduce a little more science stuff so that gardeners, I mean, it's weird because the gardening community is very diverse, right? And there are full on, biology nerds like me who both love their, heirloom varieties and don't see higher technology stuff in genetic engineering as evil. And then there's the hardcore folks who think that any unnatural thing based on their sometimes elaborate definitions of unnatural are evil. And I kind of want to hopefully help soften things for those people.
Sure. Yeah. Because, I mean, like, for me, especially professionally, I'll never force a person to be pro or anti-GMO. What I want to do is arm them with the knowledge and tools to make a decision for themselves that's based on fact, right? Especially when it comes to things like food. Like, you have total bodily autonomy over what you eat. I just want you to know what it is on an intimate level so that you don't just listen to, like, propaganda.
Yeah.
And if you finally decide saying, yeah, I don't want that in my body, I respect that. Totally respect that. But the goal is to, combat hype and fake news with actual facts. And I think that a hands-on experience would be the best experience in that sense. You not only get to know how it's made, but you made one yourself. And then you sit there and you go, what did I do? How does that impact the world? And are we supposed to be doing this? These type of moral, ethical, and philosophical questions can be had once you've actually done it, because then you know everything about it.
I'm about to wrap it up, but I just wanted to ask, I know you can be followed on Instagram at a tiny green cell, and you have a website up, but it's basically just a contact me link. Do you have anywhere else for folks to find more info on what you're doing?
So Facebook is my open lab notebook. So if you search my name on Facebook, you'll find me there, and my personal and lab life are basically one. So you'll see pictures of my dog and my plants all the time. But more importantly, I just opened up my lab notebook as a Google Doc available for everyone and anyone to read and comment on. Because when people say open source science, I really, really mean it. So I want people to actually come with me on the journey of learning and making a flower that's designed by me. And you can see all the tools being made, a bunch of ramblings about the current state of technologies. It's basically like a blog and a lab notebook, but I didn't want to have yet another platform. So I made it a Google Doc that you can just download.
And I've followed you on Facebook for a while now, and it's all very interesting, and quite a lot of it does go over my head, but it's still kind of cool to see at least a little window into that world.
Yeah, and I really do try to minimize the jargon, unless I'm just way too excited, and then I just nerdgasm all over the place. But normally I really try to make it accessible, because I don't believe that jargon's important when you're trying to be concise, but not to the general public. There's analogies that you can use and other verbiage that'll get the same message across without dumbing it down or infantilizing the audience.
Okay, great. Well, that's about all I've got. Do you have anything else you want to say before we cut out?
But thanks so much for this opportunity. It was definitely a unique one.
Oh, no problem.
Looking forward to see the feedback, whether positive or negative. Happy for it.
Great, I'll let you know. Thank you so much for listening. That wraps it up for this week. You can follow everything we're doing over at tjsgarden.com, and the best advertising we can get is word of mouth. So please share this show around with your friends, directly or via social media. Have a great day, guys, and enjoy your garden.